Welcome everyone for BioEngine webinar. This is our 72 webinar in the series. And thanks everyone for joining in this live session, although the recording is available in our channel. So now I'm going to end the registrations link because it's the closed link is now closed, and we are going to introduce our today's speaker. She is a tremendous good scientist.
Today, she will speak on a highly relevant and timely topic: the genomics of root system architecture in rice, unveiling adaptive traits for stress resilience. We are truly privileged to have her with us. Please join me in welcoming Dr. Kalyani M. Barbadikar to the virtual stage. Dr. Kalyani, all yours now.
Thank you so much, ma'am. Very good morning to all of you. And thank you, the organizers of BioEngine, Dr. Shuvo and Dr. Soma, for giving this opportunity. So if it's all right, shall we start the talk? Dr. Shuvo, is it okay?
Now you can hear me.
Yeah.
Yes. Okay.
Shall we start the talk?
audio problem is persist.
Conscious.
So what we do at the end, we'll again play this audio for your introductory part, because some part may be the missing to the correct audio setting. So
Yeah, yeah.
yes, you can share your screen, and for the audience, please be patient, listen the webinar, make your questions, put the question in YouTube live chat. I will collect for the uh, I will collect all the question for the speaker. And at the end, we give you a feedback, come e-certificate application link. Please fill the link and use the same email ID what you have done during the registration process. We match the email ID and issue the certificate. All certificate will be available after 14 days, near about 14 days, or whatever. When I uploaded, I will email to every registered participant that now you can download the certificate. So don't worry about the certificate, you will get it if the process is correct and according to our rules. So now enjoy the talk. I will collect all the questions and enjoy the question answer round. Don't bother about the certificate link, it will be provided, I'm sure. Ma'am, and please start.
Yeah, thank you so much, Dr. Shubho. So, a very warm good morning to all of you. So, today I'm going to speak about the genomics of root system architecture in rice, unveiling adaptive traits for stress resilience. So, before we begin, I would like to just take you to the outline of my talk, what things I'll be covering today. It's about the introduction, the transcriptomics, the gene expression analysis, the genomic regions which are associated with useful root traits, the functional annotations of it, variant prioritization, integrative omics and plant research, integration of omic layers for candidate gene discovery, the future perspectives and applications. The traits which I will be covering are the robust root system architecture, specifically focusing on the root length, root volume, the seedling vigor index, early establishment traits, robust root traits along with yield related traits, and the omics approaches which we'll be covering is the Mutmap or the QTL-seq, whole genome sequences, genotyping by sequencings, genotyping, genome-wide association mapping, some idea on long non-coding RNA and epigenetics, haplotyping, and integrative omics. So before I begin, I would like to introduce my institute where I am working. My institute is under the aegis of ICR, the Indian Council of Agriculture Research, named Indian Institute of Rice Research. It is located in Hyderabad at Rajendranagar. And we are having the focus at ICR IIRR for research, basic research, strategic research, as well as the coordination. Coordination in the sense of All India Coordinated Research Program. Our institute is one of the agencies which undertakes all kind of coordination related to rice, including the release of the varieties. So at ICR IIRR, we focus upon the smart rice, sustainable nutrition, and our mission and vision are also towards food security, sustainable nutrition, and the food nutrition security as well as with the food security. So we focus in our institute on many traits, including the biotic stress as well as abiotic stress, along with yield. So yield being a major criteria for us. So we do in biotechnology, we focus on marker assisted selection along with conventional breeding, the mutation breeding or haplotype based breeding, which we are just about to start or we have just started, association mapping, and new breeding techniques like genetic engineering, genome editing. So for every approach which we use, we target different kinds of abiotic stress, biotic stress, as well as the multiple stresses like BLB, bacterial leaf blight, PUE, phosphorus use efficiency, gall midge, sheath blight, brown plant hopper, nitrogen use efficiency, the root system architecture, phosphorus use efficiency, yellow stem borer, like in the pest side. Then we have rice leaf folder. For the physiological traits, we do work on strong cull, seedling vigor, photosynthetic use efficiency, then some kind of rice to grow virus or virals plus the diseases plus the pest. So for every approach, what we use, we have targeted the trait as well. So we have a very distinguished trait identification as well as the approach with which we are going to address that trait for a better climate resilience or better biotic abiotic stresses or the nutrition as well. So under nutrition, our institute has a focus on nitrogen. Apart from the nitrogen, we have a focus on zinc as well as zinc along with iron. So zinc also is a major component which is being targeted at our institute. So to be on the positive notes, our institute, along with our team of scientists and directors, sir, we have developed under the leadership of Dr. Satyendra Kumar Mangrotia genome editing line called DRR Dhan 100 Kamala, which is a breakthrough and which is in the news. This line was recently, about a month back, it was dedicated to the nation by Honorable Union Minister for Agriculture and Farmers Welfare. So this is like a very positive note. Apart from this varieties, we have also released this time in 2024, 2025 climate resilient other varieties as well. But this is a first genome edited product in India, along with one of the genome edited line from IRI developed by Chinuswamy sir. So coming on to the root system architecture, which we generally known as the hidden half, because we are not able to see this root system, and every time we are focusing on the yield, or when we go to any crop or plant or in the field, we first see that the complete yield or the inflorescence. So what is happening? But to see or to visualize the root system architecture, it's really difficult. So we generally call it as a hidden half for enhancing nutrient use efficiency. So apart from what we do for the yield or yield related traits, if we look down in the soil, what is exactly into the soil is our root, which is a main organ again, which is responsible for overall growth, the vigor of the plant, the anchorage. It provides the anchorage to the plant. It makes it to fit properly in the soil or in any media if we look like that. So it has a phenotypic plasticity, and here I have provided you a view of how this roots look like. The exactly root when we scan it and when we wash it from the media or from the soil, this looks like this. It differs from crop to crop, and also it differs in rice from genotype to genotype. So what are the factors which are responsible for making this spatial arrangements of the roots that we'll see? But this whole system is known as the root system architecture, and it is like governed by four major parameters of growth, branching, the surface area, the angle with which the root grows with the soil, and which responds to the signal in the immediate vicinity of the soil environment. So this soil environment can be water, or when we call about for the nutrients, it can be phosphorus, nitrogen, or any other element which is present. And how this spatial architecture helps to enhance the nutrient use efficiency, ultimately giving a chance for us to improve the climate resilience because of this roots that we'll be seeing in the next few slides. So this slide tells about that rice is life, and we are continuously in a mode of changing climate. We are experiencing different kind of weathers every day and the erratic rainfall. So with this, all of the system which is changing, and we are going to have a challenge also in the next 25 years. So in this way, how can we address the climate resilience or the sustainable development goal with special emphasis on the root system architecture? We are going to see in the next slides. So when we talk about the root development in rice roots are fibrous root system. They are fibrous in nature. We can see it here or have a look how they grow. They have a seminal root system, then the crown roots, in which makes the complete root system architecture. Then we have the nodal roots, the lateral roots, and the adventitious roots which develops. So this development is a complex measure. It's a complex, complete molecular mechanism which operates with a stimulus, with the presence of the nutrients which are available in the soil, as well as the stimulus which is being carried from the receptor molecules like hormones, the hormonal development like auxins, cytokinin, gibberellins, brassinosteroids, as well as the transporters, transcription factors. And these are the genes which we have listed for the root development, which goes for root elongation, crown root elongation, root emergence, lateral root initiation, and the root emergence. So if we see this figure, this gives again a holistic picture of the growth, how the root grow and rise, how is a branching pattern, and this is the seminal root. After that, when the root develops, it develops first level of lateral roots, second level of lateral roots, which forms its branching. Just put the pointer on. So which forms the branching, then how it grows, and what is the total surface area of that root. So the traits which are governing this is the length, the root length, the total length, how deep it goes in the soil, or how deep it goes in any system where we are doing a particular kind of phenotyping or whatever we say here. So next is a root volume, the total volume which is present, which is occupied by the roots in a particular system like soil or agar or any other vermicompost. Then comes the root number. How many are the number of roots which are there? Then when we take the diameter, what is the diameter of this particular roots, from the root tip to the nodal roots to the seminal roots and the lateral roots. Then after drying, this is the fresh weight, the complete biomass or the weight of the roots, the total weight of the root, and after drying, the dry weight of this root, that is the biomass, that is the biomass of the root. Then we have the surface angle, with what angle, which angle the root grows. It grows straight, it grows with an angle of 30 degree, 40 degree, 70 degree, or it is shallow or completely deep down the soil. So that again gives the root angle. Then we have the root surface, root hair density. How dense is the root hairs, which determines its root penetration ability and the root absorption also of the nutrients which is present in the soil. So these are the four growth, branching, angle, surface area, which are governed by these traits: length, volume, number, thickness, the dry weight, fresh weight, root surface angle, root surface area, the hair density, and the root to shoot ratio, which is like a derivative of the dry weight and the fresh weight. And how many parenchymas, like if we see into the histology, we have the parenchyma formation as well. So these are like the root angles what we are talking about here. We can see how IR64 grows. It is deep, shallow, like that. So these are the basic how root phenotyping works, or which are the traits which contribute to our phenotyping. So how to see these roots now? The ways to see the unseen or the hidden half. So there are several methods. How do we do root phenotyping? Specifically by the root excavations and trenching. We have mini rhizotrons, shovelomics, the polythene bag method, rhizoboxes. We can grow them in the hydroponics or aeroponics, or in the Petri plates, filter papers, transparent growth media, X-ray 3D imaging, or in the transparent growth medium. Because when we are trying to address the issues or for the development of the new varieties or mapping the genomic regions, we should we should have what are the methods of its phenotyping, which trait to focus, and how to see the trait, how to phenotype the trait of interest, so that that trait of interest we can take ahead for the genotyping, or to correlate the genotype with the phenotype, and then the map the genomic regions which are underlying those traits, and how these traits then ultimately goes into the breeding programs or goes into the biotechnological applications of molecular mechanisms. Understanding those mechanisms depends on how precisely and how with reproducibility we do the phenotyping. So these are some of the field phenotyping methods. like Shovelomics. This is for the root angle. Mini rhizotron. Mini rhizotron is an instrument which has a sensor which will allow us or the user to understand how deep these roots are going down the soil. There are greenhouse methods. Every facility or every institute, per se, every lab has its own methods according to the space availability, fund availability, according to the systems, expertise, collaborations available. These phenotyping methods can be like either they can be standardized or they can be adopted when we are doing any kind of root studies. So these are the greenhouse methods. So this is a very special field facilities which are available at our institute. We have a normal irrigated transplanting field, which we call the irrigated field. It is available in our institute. These are our institute photos for our field. Apart from that, we have aerobic field, that is direct seeded field facility, which is available here at Hyderabad. So irrigated field facility is a transplanting facility where we grow the rice seedlings on the bed, and after 21 to 25 days of its growth, we transplant it and give it a flooding, saying that the water is always available 2 to 3 centimeter or 4 centimeter above the ground level. In contrast, here as we can see very clearly, this is water is standing here and there is no water here, or this is a aerobic condition wherein limiting water condition is available. The irrigation is given, flash one irrigation is given at one time, and only soil based moisture is maintained every time. There is no standing water here in this condition. And this we follow the direct seeding method here. That is, the seeds are not the seeds are directly placed into the soil and there is no transplanting done. So the seeds are directly sown in the soil and then observed for its subsequent growth and selections or any kind of phenotyping. So for other nutrients also, field phenotyping facilities which are available at our institute, we have a very well recognized low N and low P field facility. In low N, that is the nitrogen level, there is a zero application, that is nitrogen fertilizers are not applied. They are applied at 50% recommended dose of nitrogen for rice and the recommended dose of nitrogen. So anytime for nitrogen uptake efficiency or nitrogen use efficiency, we have a dedicated field facility wherein the genotypes or the rice lines can be screened. Similarly, for low P, that is the phosphorus, here in the low P soil, recommended, there is one recommended field. Another, in contrast, we have low P wherein no phosphorus fertilizer is applied and the phenotyping or screening for plants, for root, for yield, and for other physiological parameters are being studied. So towards going to simplifying the root traits, the methodology which our team here follows for root traits are the polythene bag based phenotyping, which is done in a controlled condition with a specific amount of soil in the polythene bags. We follow a methodology of 60 to 80 centimeter polythene bags. So up to 60 centimeter, we fill the soil. This soil, when we go for very transcriptomic studies, this soil is an autoclaved soil system in which 15 kg per polythene bag is filled, and we are doing phenotyping for roots under irrigated condition. Irrigated here means we keep 3 to 4 centimeter water levels maintained in the polythene bags. And for the aerobic one, that is a dry condition, we have holes in the polythene bags which doesn't allow water to stay, and minimum irrigation with measured water is always given, and the soil moisture is maintained. And this is a kind of phenotyping. In detail also, I have one slide for this. And this is how we phenotype at different stages. So different stages of rice is one phenotyping. For roots, we are doing it at the seedling stage. About from 7 days, we start 7 days, 14 days, 21 days, and sometimes even up to 45 days. And then panicle initiation stage, which differs in rice genotypes from 80 days to 90 or 90 to 100 days, like that. And this is how we phenotype. This picture I have kept, which is a When we started this work around 8 to 9 years, so these were the breeding lines which were very kindly shared by breeders of our institute, and we collected these lines, then started slowly with the standardization of the phenotyping in the polythene bags. And then this is one of the facility which is again available at our institute for WinRhizo. This is a WinRhizo Pro software. So when we scan these roots look like this. And then we go for its volume, the length, the root diameter. So these are another root laboratory methods for rhizoboxes. And as we do the CT scan, this is one of the methods where CT scans are done to understand how much is the root growing deep and what is a root growth. So these are some of the tools which are used for root phenotyping. The WinRhizo, which is maximally used, and this has an upgraded version also, which allows us to understand even the baby roots or the hair, the very fine roots as well. Another software is RhizoVision Explorer, the digital imaging IJ software, Rhizo biometric software, RhizoVision Crown, GT roots, and dynamic roots. So some of these softwares are freely available and some are available with the scanning of the scanners, a particular scanner and a computer and a tool. The software is like linked to it, and some are paid versions also, but many of them, they are like free versions which can be available online. So now going again back with the main topic, like how do we connect the roots with the climate resilience or with the stress condition, or how we can harness the traits, the root traits, for maximizing the resilience in context with water, nitrogen, and phosphorus. So these three, water, nitrogen, and phosphorus, will be my main focus. So for climate resilience, from transplanted rice, which requires more amount of rice and labor for transplanting, we are slowly heading towards the direct seeded rice, which is a limiting water condition. And in this limiting water condition, how roots allow us for selection, for the breeding, and how we understand the molecular mechanisms which underlie these particular root adaptation, because roots are very plastic in nature, so they do exhibit plasticity. So which kind of root traits are suitable for our limited water condition? Then similarly for nitrogen, when nitrogen fertilization or phosphorus fertilization are given in a limited dose, how we can harness this root traits, so that along with the root traits, when we focus, we also have a better yield, because yield is a major criteria where our stakeholders or our farmers are interested into. So whatever we do for the betterment or for climate resilience, it has to be very well, it has to go hand in hand with the yield. So maximizing the yield under limited nutrient condition or limited water, nitrogen, phosphorus conditions. So in this diagram, we can see that how this roots help in the uptake, then how it goes with the assimilation and to its maximum utilization, so that it expresses itself, whatever in the roots, the mechanism is followed, it, it goes up to the grain filling and to the yield. So adapting the underground, again, when we see which are the traits here, for drought condition is different than direct seeded condition, because direct seeded conditions are maintaining aerobic condition. Direct seeding is an establishment method and maintenance method is aerobic, which is water limiting condition. So drought is like complete absence of water for few days. So for drought condition, which is a preferable root trait for selection or for phenotyping, and which can be considered for our breeding approaches or for any molecular approaches. It's like the deep rooting or the steep root. Then thick roots for soil penetration. For nitrogen, it should have dense lateral roots with more number of or greater number of root hairs and root surface area, and for foraging that nitrogen as well. For low phosphorus, it should be the shallow root angle, the fine roots, long dense root hairs, and high root exudation for phosphorus solubilization. For direct seeding condition, because it is directly we seed it, it should have the ability to establish at a faster rate, and it has to compete with the with the weeds. So for this trait, we have long mesocarpal or coleoptile length, which is also known as the seedling vigour index. The seedling vigour should be higher, and it should have a higher number of nodal roots for anchorage and rapid growth, along with a better deep system, but the high root volume or surface area as well. For the flooding condition, for which rice is very well adapted for flooding condition, we have shallow root system with aerenchyma tissues, and it should be tolerant to under hypoxia conditions for the formation of roots. So these are the specific traits and the preferable root traits for breeding, which I have just given in a nutshell here. And now we'll see how do we address this, or what are the molecular mechanism which we have worked upon. So when we started understanding the root system, we thought like, okay, we'll go with the aerobic and anaerobic condition, or the irrigated is known as the anaerobic conditions, aerobic conditions. And then we thought like we'll study how the roots, or which is a molecular mechanism that operates under under the flooding as well as under the aerobic condition. So we selected meticulously two genotypes, BPT5204, which is Samba Mahsuri, a very well mega variety in the southern regions of India, and CR-202, which is released by CRRI Cuttack. So this is our aerobic check even in our all India coordinated research projects for aerobic rice. So here what did we see after selecting these two genotypes? We thought that how the roots in these two behave, or what is the adaptation that makes CR aerobic or high yielding under limited water condition against BPT5204. So for this, we studied two tissues. One is a shoot tissue, because we say that panicle initiation, physiologically, the panicle initiation stage is very critical. And after this, maximum of the nutrients or the energy is diverted for panicle development. So these panicle development first initial tissues we have chosen as a shoot tissue to understand what happens here. And simultaneously at that time point, we have selected the roots at the panicle initiation stage to understand the aerobic adaptation, modulations in the roots, the molecular mechanisms which in the roots that uptakes the water. So for this, we took the approach of transcriptomics, and we did RNA sequencing of the root and RNA sequencing of the shoot. We went ahead with the differential expression of genes. Then after we got genes, we also did the KEGG geo-enrichment. We sorted apart and resorted to the alternative splice variations for understanding the molecular mechanism, that is there any alternative splicing mechanism operating. We mapped all the genes with the QTLs which are reported for rice and conducted this experiments. So the results were quite interesting. We thought whatever we anticipated, whatever we had hypothesized, that under the aerobic condition, how our aerobic adapted genotype behaves may be attributed to the roots. So here we have seen and we have observed, we recorded that the root related genes are highly upregulated in the aerobic adapted genotype. And these are the very specific set of genes which were highly upregulated and differentially upregulated in the aerobic adapted genotype, like in organic phosphate 1,6, the cationic transporter HKT8. We have a sweet 16, apart from that sweet 3. These are the sugar transporter genes. Metal nicotinamide YSL2, YSL13, and phosphate transporters. These are the genes which are related to the nutrient uptake or the nutrient use efficiency related genes, along with the expression of transcription factors like MATS15, which we have seen exclusively in the roots, and in the shoot we have MATS5, 6, 4, 5, 6, 7, and WRKY6 transcription factor. The major point what we observed, wherein we have deduced this molecular mechanism or we proposed a pathway for aerobic adaptation, was that aerobic adaptation, it mimics drought, but it doesn't follow the complete drought mechanism. Certain genes are expressed, but not up to that level. It is expressed only up to a specific level with the upregulation of these nutrient uptake genes. So we thought like when we are doing the hypothesis, then there are the sensor molecules. Once this sensor molecule sense the availability of water, then we have the hormonal signaling. And we observed in the roots that brassinosteroid signaling molecules were present. And brassinosteroid, since the genes were present there, we thought that it is a conglomerative effect of auxins, ethylene, and brassinosteroid. And these induce the specific transcription factors like the RAP16, C, B, D, then the DREB, basal helix loop helix, followed by a major transcription factor of MADS15. MADS15 is involved in the transcription modulation of roots as well as the floral development at peculiar conditions. So at this particular point, we found MADS15, this molecule of MADS family protein in roots, then followed by PST 1.6, the MDR like ABC molecules, and VIT, the vacuolar iron transporter homolog 2, along with SWEET3A, peculiarly upregulating exclusively in the aerobic adapted genotype, followed by aquaporins and metallothionein. So we also observe metallothionein genes MT2C, which we propose that they contribute to the aerobic adaptation. And with this, we have also all these genes we have validated in the roots and shoot at two conditions of seedling as well as the panicle initiation stage. And we have also published this research. Now for the validation of the genes, for the accurate measurement and accurate reproducibility and defining that these are QRT experiments or quantitative real-time experiments which we undertake for understanding the gene expression studies, we should have a set of reference gene. So we also identified the set of reference genes for QRT PCR gene expression. So why did we take up this study was, once we are studying anything for the roots, specifically for the aerobic or anaerobic condition, and we begin these kind of studies, it is very, very essential that whatever validation we are doing, that should be foolproof and it should be reproducible, accurate, and at a time it can be followed by everybody else in other labs or in our labs also. And to set up these reference genes, we conducted a separate experiment taking two genotypes which are adapted genotypes for aerobic condition and two anaerobic or flooding adapted genotypes like BPT and Birsadan 111. Birsadan 111 and CRdan 202 is for aerobic adaptation, and BPT and MTU 1010 is for flooding adaptations. So here we conducted in-depth studies for QRT, wherein for these two aerobic and flooding condition at 3 stages of seedling, tillering, and PI stage, we collected the tissues and then took 10 reference genes and evaluated in 6 experimental sets that which of these 10 reference genes, because reference genes are the genes which are like continuously required for a proper growth and development, and it is tissue specific also and stage specific also. So at a time, may not all the reference genes will suit one particular condition. So with this hypothesis, again, we identified the most stable reference genes And these most stable reference genes we identified from a set of 10, we came down to a set of 3, and we did it with 2 to 3 softwares along with the M value and the V values, which are the golden standard for conducting any quantitative real-time gene expression studies. We use genome, norm finder, best keeper, and ref finder, and came to a conclusion that a set of 3 genes, membrane protein, tumor protein homolog, and express proteins, even if we use 2 out of these 3, it expresses stably across a set of root and shoot tissue, specifically for this aerobic and anaerobic condition. And now in our lab, we are using this set of genes, and they are working quite well. And this study also we published. Now, coming from evaluating only the 2 genes, again, we gave it a thought, why not to take more? Because there is a lot of diversity in rice according to the geography, ecosystem, ecology, and the way we are growing the rice at different altitudes, like right from the flooding, we grow it in the upland, in the flooding condition, in hills we grow, in down sea, deep sea also it is grown. So now, taking that into consideration of wide diversity, we thought like, okay, we'll go with an approach called genome-wide association studies for root and yield related traits under aerobic and irrigated condition. So we got certain leads from studying these 2 to 4 genotypes which are adapted for root as well as to the irrigated condition. So we thought like, why not to take a bigger panel of genotypes with high diversity, diverse line. So we formed a panel specifically for the root traits. Here again, I'll acknowledge all our breeders who gave all these lines for the initial root studies under the field conditions, under the polythene conditions, polythene bag or the controlled condition. And this set of genotypes, we also checked it for the yield related traits or yield traits under the aerobic as well as the irrigated condition. So this was a complete experimental setup for the yield traits, and this is for the phenotyping for seedling, root traits at seedling as well as panicle initiation stage. And for the yield traits, we have evaluated the yield traits in our institute ICAR-IIRR as well as one place called Dhadesuguru in Karnataka, because this was a part of my PhD students' one work. So we evaluated at 2 systems, as well as we have taken all these readings 2 years, because 2 to 3 years of data across the different conditions is necessary for conducting any kind of genome-wide association studies. So we have considered that at the seedling stage, at the panicle initiation stage, the yield at the reproductive stage. We have also conducted the seedling vigor index, that is at the seedling stage, the root studies or the root length, root volume, root dry weight, fresh weight in the polythene bags in the glass house, as well as using the paper towel method. So this is a complete material and methods or the setup or a flow diagram of the experiment which we did. And the genotyping here was done using the SSR markers. And then we could find out the structure of the population. What is a population structure? Because initially we had taken the lines, land races, accession, wild derived lines, the tropical into japonica lines, and we evaluated completely for its root traits. And this was a structure. This is a genotyping. And then we could associate it. We use Tassel software for this, for understanding the marker trait associations. And simultaneously, when we did all these experiments, we also found the genotype environment, the stability of the genotypes in different environments. And we could come out with some lines, land races or accessions, which were better in the root traits as well as in the yield. So for seedling vigor index, certain land races like Langpao, this is one of the mutant of BPT called TI-128. Then we have the root traits, the root length, which was better. The genotypes like Morang Phao, JBB, the NPK45. The root volumes, for root volume, which genotype is better? So all these lines we could deduce like, okay, these are the better accessions. And along with that, we found the marker trait association. So this is a phenotype, and this you can see the how robust this root is with the volume and along with the length and how deep it goes. So we evaluated by this method at the seedling stage as well as the panicle initiation stage. And these are some of the robust or better root system lines. This is a genetic diversity of among this 118 GWAS lines, which we could get. And this study was for both, like we did at the root phenotyping, completely the root traits like root volume, root length, then root dry weight, fresh weight, along with the panicle traits like the yield, how much was the yield and yield related attributes. So with this two, we could come out with a certain set of genes like oxygeniflux carrier protein, which was present in the both the tissues and had a marker trait association. So with a better marker trait association, we came up with one HbAF2 basal zinc transcription factor, nodulin-like protein, one amino acid transporter. So for all these, we have marker trait association with more than 2 or 3 traits at a time. That is with the root as well as with the yield. So patatin, and here we came up again with, we observe one MADS 80 transcription factor box, one protein. And these are the markers which are associated with it, RM5501. RM5501, then we have 410, and you can see like with how many traits it is, how many genes it is correlating in terms of marker trait association for more than 2 or 3 traits. So this work also we published. So now coming on to this, like we had this panel of genotype. So this GWAS panel, we thought, okay, if it is better performing under the limited water condition or the aerobic condition, can we again check it out how it behaves under the limited phosphorus condition as well? So we took the all these phenotyping under low-P field as well as with the genotyping. For few genotypes, we have done the low-P field screening in our institute in itself, and then we did the genotyping using the phosphate uptake 1, because this QTL is very well known and very well characterized QTL, and there are diagnostic markers for low-P. So we came up with this study. We did genotyping and then we did a limited phenotyping under low-P plot, and we came up with one land race which we are going to register it also. It has a very good high root volume and yield under low-P conditions. So this, like this, we came up with few lines which gave, with the genotype marker, it gave good understanding of how there is an interplay of water use efficiency or water uptake with phosphorus uptake. Then one line which we have identified in these 3 lines, they are in our All India Coordinated Research Project trials as well. And these 2 lines we have taken further for further understanding why this robust root, because we are getting the phenotype. So how do we get inside and dig deeper into the molecular mechanisms which are operating that makes this as a better candidate for nutrient use or better climate resilience for the next breeding programs. So we took one line, and one of these line, again, we mapped the genomic regions. So here we have taken advantage of our collaborative networks and All India Coordinated Research Project. After developing this particular mapping population, this is a biparental mapping population which was developed, and this was phenotyped at 4 locations across all the zones. And this is a field view under aerobic condition, how this was grown. And here we have done the genotyping for this biparental population using 1K RICA genotyping, which is a 1000 rice customized base array genotyping. And the phenotyping was done. These are the various locations where these mapping populations were phenotyping. We phenotyped at our institute, then followed by one institute at Karnataka called Kathalgiri, in Karzat, RARS Karzat, Rohtas, which is at Bihar Sabaur, and Navagam in Gujarat. So we same mapping population, we have done phenotyping at 5 locations, because genotyping was done at our institute. Then we did almost 2 years of phenotyping at this 5 location to understand how these genomic factors and environmental factors interact with each other. And then we came up with the mapping. mapping these genomic regions. We have also conducted the root phenotyping for all these mapping population, the candidates like over 100 we had, and this was the root phenotyping. So in aerobic conditions, under control conditions also, phenotyping was done for roots. So this was done for yield, this was done for root, and again we could map the genomic regions, and we came up with 4 or 5 genomic regions. This work is yet to be published, but we could map certain regions which we had already found in our other studies. So now this was biparental mapping population. So we thought like one of the line, which is a mutant of BPT5204, we thought like we'll go dig deeper into it specifically for 2 traits of root volume as well as the root length. So for this, we have used an approach called mutation mapping, which is a bulk segregant analysis based approach, wherein the two parents, the two lines, the two parents, two lines, two genotypes are crossed. One has a specific trait, the trait of interest, and it is crossed with another line, which may be a popular variety, and a popular variety or a genotype. And then it has to be phenotype, but this phenotyping is done at the F2 or sometimes in the F3, and it is bulked. The extremes of the phenotypes are bulked together and directly sequenced. Typically, it is known as the whole genome resequencing process. But here, along with the parent, single line or genotype, the bulks are also sequenced, the complete pool. Like here, we have done the phenotyping for root length. So this is a bulk, which is for root length and root volume. And this is a bulk for low volume or low length. So this is one bulk. So 15 plants of each we have pooled in equimolar quantity of DNA, and then it is subjected to resequencing. So here, this particular approach of Mutmap or QTL-seq. QTL-seq we say because we go back to the 2 genotypes and then take genotypes for reference and map those. But this harnesses or it exploits the approach of BSA, bulk segregant analysis, at an earlier generation like F2 or F3. And again, we go for mapping these genomic regions for the particular trait based on the complete pool of one, which consists of 15 or 20 individual plants. So when we have done this Mutmap analysis, we did the genetic mapping, and this particular mutation mapping method is depending on the SNP sequence or SNP, which we call SNP indices. These SNP indices are the one which are responsible for the mutation or which are responsible for the trait of interest or change that drives our trait of interest. So we found that in genomic region of 12, we had 2 regions, 3.1 to 3.7 MB, as well as from 18 to 20.85 MB we have mapped. So these SNPs are the SNPs which are responsible for conferring root volume or a better root volume or a higher root volume and root trait as compared to the parent, to the other parent. And this we could map at 2 locations on chromosome number 12 as well as chromosome number 2. On chromosome number 2, it was 23.18 to 23.68 MB. So these regions are like very, very narrow regions, fine regions, which can be further deployed for better root length and root volume in the breeding programs. So once we are doing this, we are going from the big genomic region, we are going to the genes as well as the SNP, the variant, the single nucleotide polymorphism which is present, which may or may not be responsible for conferring the trait of root volume or root length. So for this, we again validated, to understand this, we validated these SNPs. And here we can see this is S4. We designed competitive allele specific markers on chromosome number 12. There is a transition of C to T, and this is a location of this SNP, which is located in the 3' UTR of a gene which codes for a protein kinase domain containing protein. And as well as for the second chromosome, we have selected one primer. This S6 is nothing but a Cas primer. And here also it is C to T. And then we tried to design, it is present in the upstream of a gene called chromosome condensation protein. So we designed to validate these SNPs. We designed the CASP markers, and these are the results of our CASP marker, wherein we can see that in CASP competitive allele specific marker, when we run it, we are running it on a DNA. And when we run this on the DNA for validation, a large panel of genotypes or mapping populations are deployed in such a way that some of these are already phenotyped for a better root system, some for a low root system. So we form a panel, and all these, so each dot here is one genotype. One circle is a genotype over here. And using this CASP markers, we found like maximum correlation for these three SNPs which were designed. And we propose that these three genes along with the SNPs may be playing a crucial role for conferring the root length as well as root volume. So we had validated these on a panel of around 70 genotypes contrasting in the root length and root volume. And these are the CASP markers. So this study also we published last year. So when we go now we understood that this is a SNP which is co-segregating or correlating with root volume and root length. So now how to go about it and check the expression. So we also checked the expression of these genes in our two better genotypes adapted for rice or having root volume and high root length versus BPT5204, which is our popular variety. And we could see that two of these genes, protein kinase containing domain, were highly expressed, as well as the regulator of chromosome condensation domain containing gene was also expressed better in our genotypes showing the better root volume and root length. So in this, we propose that these two genes, protein kinase containing genes and chromosome condensation domain gene on chromosome 12 and 2 do play an important role for this particular trait of root volume and root length. So again, we are like narrowed down to these two genes along with two CASP, which can be deployed further for breeding programs for root volume and root length. So now again, coming to another aspect of root regulations, when we are telling like, okay, this is a molecular mechanisms, or these are the genes which are underlying for the better root and root volume, we again thought like any other mechanism of specifically the epigenetic mechanism, do they have any role for roots under aerobic adaptation, or how do they impact the adaptation specifically for the roots? So here we did with long non-coding RNA, which are more than 200 nucleotides and having no or very low coding potentials. Low coding potentials are like they are non-coding RNAs, and these long non-coding RNAs, they do play an important role, very crucial roles in the development, stress tolerance, as well as in the level of the expressions. They act as miRNA, the microRNA mimics, as well as they participate in the complex regulatory networks. So we thought like, okay, these LNC RNAs, long non-coding RNAs, which are non-coding, but they may have do they have any kind of expression control or regulation control? So with this hypothesis, we tried to do in the same genotype, the roots, transcriptome analysis or RNA-seq for both mRNAs as well as long non-coding RNAs. So we have also published one small review on this long non-coding RNAs and how do they mediate the stress response specifically for the abiotic stress tolerance in plant. So with this hypothesis, like, do LNC RNAs play a role in root development under aerobic condition? We designed this experiment, did RNA sequencing, and we came up the better root system, roots in that genotype, which are the upregulated genes, along with the upregulated genes or the mRNAs, which are the upregulated long non-coding RNAs. And interestingly, we found a plethora of long non-coding RNAs which were highly regulated or highly upregulated in the better root system genotypes, having robust root system in terms of root length and root volume and dry weight. And we came up with these 4 to 5 candidates. This XLOC mentions the long non-coding RNA because they are non-coding. So, and now we are relating these LNC RNAs with the mRNAs and checking out whether there are any candidate mRNAs which are upregulated with a consequent downregulation of LNC RNAs. And we are making these kind of networks also. This work is in progress, but I wanted to show this as a point, like for the roots, apart from the genetics, epigenetics may also have certain roles or regulations like in terms of long non-coding RNA expression. So coming on to the next point of haplotyping, that which are how do we harness this haplotypes? Haplotypes are set of the SNPs or set of nucleotides which are inherited together. So under this aerobic condition or dry direct seeded conditions, do we see any kind of haplotypes which are present? For better understanding of the haplotypes which can be deployed further for haplotype based breeding, this was our one project funded by NASF, in which we had done haplotype diversity of 3K, that 3K indica lines. This is a big panel because rice also has 3 to 4 big panels, already genotype panel. So we had selected only the indica lines for this, and we did genotyping for dry direct seeding as well as for nitrogen and phosphorus under the limited conditions. And here are the results. And here also we did at the seedling stage as well as the panicle initiation state the root phenotyping, because here we did phenotyping and genotype data is already available on a 3K SNP-seq panel. And then we conducted the GWAS for early seedling vigor traits as well as the yield traits. So these are the root traits. And then every time when we are doing this exercise, we are also finding certain genotypes which are better performing across all the locations, because once we have these kind of genotypes, we can use it as donors for our breeding program. So superior genotypes also we are identifying. And here when we have done the phenotyping for roots as well as the grain, as well as when we have done for the yield, we could get some of the common MTAs, the genomoid we have done. We got common MTAs across the locations. So across the locations we got on chromosome number 4, and here I have just put one representative picture that how we are getting common MTAs. So generally in all the haplotype based methods, when we are getting common marker trait association across the locations, we say that yes, they are the one which can be banked upon further for introgression purposes in haplotype based breeding. And along with haplotype based breeding, we also saw the candidate genes for haplotypes. So based on our earlier research from our institute only, we have selected, these are some of the studies which were published from different groups. This is a study where we have done from integration line from Oriza Nevada. This is from Dr. Divya Madam's group, and this is from Dr. Giri's group. So whatever we had done from our institute along with the published data, we thought that we'll go with the haplopheno analysis for certain genes which we have identified from the haplotypes here or from the GWAS. So with that, we could get certain genes, which also 2 or 3 genes which were common from our earlier analysis of Mutmap and QTL sequencing. And this gave us a confidence that yes, when 2 or 3 haplotypes of the genes, they are getting repeated on the basis of expression as well as from the genomic mapping, we are more sure of what the candidate genes can be further deployed. So this work is also unpublished. So this is for the better root system architecture for nitrogen use efficiency. So this is a project which is led by our head madam, Dr. C.N. Nirja ma'am. So this we done, as we have the 3K panel, we have the Bengal Assam OS panel. These panels are diverse genotypes which are already phenotyped, and they are like in these are like the small association panels or mini panels which are available with already genotyped. So any one of us who really wants to dig deeper into for different kind of traits, we can have this lines procured and then we can do the genotyping. So in our study, we have done Bangala-Assam OS panel lines for nitrogen use efficiency, and under low nitrogen condition or graded nitrogen plot, how are these roots? So we have these are the root phenotypes or the diverse uh, architecture how we are getting for each Bangala-Assam OS lines. So we can see like which genotype we can select further, or how this root system architecture contributes for better adaptation under nitrogen conditions. Yeah, with the deeper rooting or with a robust volume like that. So some of these lines we have selected and the work is under progress for GWAS analysis. So we thought like, is there any cross combination or is there any crosstalk between the phosphorus and the nitrogen? So for this, conducting this kind of studies, we have taken our already identified genotypes of like PHO12, PHT16, YSL. Remember the earlier nutrient use efficiency genes which we had identified from our expression studies, from QTL-seq, Mutmap studies, and other GWAS studies. We have taken few genes and then validated it under different gradients of nitrogen as well as phosphorus. And interestingly, we got that certain genes like NAS, PHO, MT2C, PHT 1.6, they are expressed under low P as well as low nitrogen conditions. And there may be some kind of crosstalk mechanism which exists in rice, specifically in terms of root adaptation. So we have done this study, and these are the root phenotypes which you can see how one or two genes which are adapting here, and these are the genotypes with better root establishment or better root seedling vigour index. So this is a recently published study wherein we have integrated our molecular network which are responsible for nitrogen use efficiency. So we have already identified through RNA sequencing or transcriptomic studies and nitrogen use efficient genotypes and non-efficient genotypes. Like what are the crucial genes which are up or down regulated as compared to the non-efficient genotypes for nitrogen uptake. Then this data of RNA-seq we conducted one separate microarray sequencing or microarray profiling of root and shoot at the seedling stage of two genotypes, one efficient for nitrogen and another non-efficient for nitrogen under hydroponics, as well as we have conducted one micro RNA expression profiling of the panicle tissues under low nitrogen condition and sufficient, like recommended nitrogen condition. So here we can see these are the two genotypes, Prasanna and Varadan. And you can see this particular root growth, how it is enhanced under low nitrogen. The root is having a high volume as well as the more length and lateral roots as well. So this we have subjected to microarray, and we got certain genes which are upregulated like GARP MYB, early nodulin genes. Nodulin genes, remember we found it with the marker trait associations in our earlier studies under for root under low water conditions. Then we had an also enhanced expression of BR1, which is a brassinosteroid insensitive protein, IAA, POP. These are auxin related genes, and the dove genes also. And here we came up to a conclusion by integrating all these three data sets in the roots as well as in the shoot at two stages, in the seedling stage as well as the reproductive stage, that there is a coordinated hormonal and nutrient signaling response which contributes to the complete adaptation. And then we came up with a set of genes as well, which we are taking it further in our another projects for digging deeper into it and understanding the mechanism and how these genes are playing role under low nitrogen conditions. So this is one of the genotypes which we had identified from the genome-wide association panel study. This is an EMS mutant of BPT5204, Samba Mahsuri, that has been registered for a robust root system architecture. These are the lines which we have kept it for a comparison. These are the WinRhizo pictures, the scanned images, and this is registered with NBPJR New Delhi. One of the lines which we have identified or which we have been studying for the past 6-7 years, it has been released as a climate resilient, low soil, phosphorus tolerant variety. This line also has a robust root system architecture, and this has been released last year as DRR 73, and it was one of the line which was dedicated by Honorable Prime Minister Sir for climate resilient agriculture, specifically in rice. And this is released for the states of Karnataka, Andhra Pradesh, and Telangana. So how different approaches we are using and how these omics technologies can be applied for understanding this nitrogen, phosphorus, or water uptake efficiency. There are various approaches, and this is one of the book chapters which we have published from our institute for this. We are also maintaining one database of the resequencing data and whatever this Mutmap data, nitrogen data, RNA sequencing data, individual whole genome sequencing data, we are maintaining it at one place. And this we have done. This database is developed in a project of bioinformatics called DBT NNP projects. And this is all our team which is maintaining this database. And this is actually one interface which will take again to NCBI for downloading the basic data. But whatever NGS data or which we are utilizing for the roots, for the aerobic, or for nitrogen, they are all available with one click. It can be accessed from this database. So the take-home points which I prefer or which I want to highlight here is root system architecture is important. It is a very, very crucial for stability, flexibility. It has plasticity. It can be definitely deployed, and it has a potential for sustainability, specifically in terms of low nutrient conditions like nitrogen, phosphorus, water, which we are focusing here. So, and these can be related very well to the root system architecture and how best we can phenotype and select under these conditions. Apart from this selection of the roots, how it relates to the yield, that also has to be always taken care of. And whatever reported genes are QTLs we are having, it all depends on which kind of characters, whether we are characterizing it in the soil for limited water condition, nitrogen, phosphorus. So they have to be like deployed with very carefully. They should be deployed and mapped also. And it is very crucial to select and breed for root system architecture that does not compromise with the yield, because yield is our major parameter. Every time these roots have to be connected with the yields as well. So we'll typically, it is like yield under low nitrogen or yield under limited water condition, yield under dry direct seeded conditions. And always the extreme line with the phenotype may not work so with such a good efficiency, because it is like a holistic approach. For roots, what my take home message is, when we are studying only for the complete depth or the length, along with the length, a substantial amount of volume is also required when we consider direct seeding or maintaining aerobic conditions, along with the yield. So we have to take we have to consider early vigor, better root length, better root volume, and an optimum of all these traits, which will holistically contribute to a better uptake or efficiency. Our future perspectives for these studies are, we should focus is needed for identification, mining, or targeting the meta QTLs for nutrient use efficiency for deployment of QTL regions in breeding programs, because there is some kind of hotspots can be identified. We are also in a progress of doing and integrating all these QTLs which have been mapped till now, and how we integrate this together for a better nutrient use efficiency. And this has to be deciphered. A fine mapping of these regions can be done or needs to be done. And in addition, we have to go with the targeted breeding approach, which is aimed for cultivation of specific crop cultivation, specific areas, area wise, or pocket specific according to the status of nutrients available in the soil. And genomics approaches like GWAS, haplotype based breeding, can be deployed right now in the light of especially the next generation sequencing and the accessibility of next generation platforms which are available. And we can go for an integrated approach of GWAS, RNA-seq, or functional characterization using any techniques or new breeding technologies to further dig deeper into the SNP, the gene, and how it is associated with the trait of interest, particularly for root traits. And here I acknowledge all of our team, our director sir, our head madam, all our sir, earlier, all the principal scientists who have helped us, all the heads of the departments, because this is not an individual work. This is a complete long work of big team. We have a team of research scholars, our collaborators, our scientists, the complete IRR family, because for the root traits, it is important for physiology, for biotechnology, breeding. So it is an integrated work, and I acknowledge all of them, all the funding agencies, and especially the organizers of this talk for giving me this opportunity to present our work of 8 to 9 years here at this time to a large set of audience here. So thank you, BioEngine entire team and Dr. Shuvabrata for this opportunity. Thank you so much.
Thank you. Thank you, ma'am. It's great, and many appreciation I already received. Maybe their PhD students and all, because their questions is like that software name and all. So yes, I have collected few of questions from the live session. So yes, it's a great presentation, and it has everything. Means the basic informations, updated informations, and link of your publications. So it has whole dimension, and your talk will be recorded here, available as a recording. So anyone can use it in their future.
Sure.
So, one or two questions we have already collected from YouTube. So that is one is before going that we gave some time for others audience for more and more questions. So we do the your interview sessions and little bit discussions with me and you. So people are engaging with us and they get their questions.
Yeah.
So we are going to stop your. Uh, you can stop your screen sharing, ma'am.
Sure.
Okay. So what we have, I have gone through, because sometime I be busy to do the YouTube and all. But you have mentioned that you have a nitrogen-free field. That means for many years there is no use of fertilizer, or how it can be maintained there. Means
No
it.
additional fertilizers are added, and it is a gradient plot, complete segmented plots, and we are maintaining nitrogen gradient there.
Okay, and it's there is any chance of drain out the nitrogen, or it's not happen, means the how it protected, means there is a.
That's what we have.
Okay.
Clear cemented blocks.
Okay, cemented blocks, huh?
Cemented blocks. So it
Mm,
goes down in meters as well.
mm,
So it protects the water coming from one gradient to another.
mm.
So
And
perfect partitioning of it.
okay, and in your lab, any model plant like those root are easy to handle or can be grow in the lab? Is it used as a model like Arabidopsis, or not? You are
Yes.
fully uh, in a in a rice. Okay, so
Also,
uh,
phenotyping we are doing using paper towel methods. We are
hmm,
doing glass methods, polythene methods, as well as the field methods.
hmm,
So depends on which stage we want to take it for the phenotyping. Like if you want to phenotype for seedling stage, we are doing it at 7 days after germination, 14 days, 21 days. So we are using paper towel method also.
hmm. You have already mentioned the epigenetics, means non-coding RNA involvement. But any microbiome studies, means anything? Uh,
Microbiomes also we have started and we are doing it in two or three of our projects. One
hmm,
one is in the BNF project, biological nitrogen fixation, one
hmm,
isolate concept project we are working on it.
hmm.
Right? Like what it
Yes, it's a very initial stage, so that's why you're not showing it. But yes.
but very interesting results
Hmm.
what we are getting. So we are
Hmm.
applying the peculiar
Yes, because there is a high chance of secretion of auxin and others hormones by the others microbes in there. So maybe there is a high chance to get something which is also controlling the architecture of root.
is true.
So because root is a very fascinating world, but very hard to work. Means those are people are working on roots is a very much challenge to just isolate root and imaging. That's the simple thing, but it's a very tough in that way.
Phenotype
And
tedious.
yes, and in bioengine we always focus the root. Means there is maybe 3 4 talks on only roots. Your maybe the fourth talk in the root in bioengine because it's matter. So my personal interest, I have seen DRAVE in your presentation, but no RD29 related thing in your findings. So what will the cause? Because is it more in salinity, not in drought in that way?
No. It is morely like that's what we got some DREB1 gene and
Hmm,
all like promoters, but we did not get those genes which are expressed peculiarly for drought.
hmm, hmm, hmm,
Like, this is like quite like we thought like yes, this is a mechanism because it just mimics. Because under drought it is a complete withdrawal of water, but in aerobic conditions we are not withdrawing. It
hmm.
is like a limited water. It is moisture is always maintained.
Okay.
So that's what we hypothesize. Like the nutrient uptake genes
Hmm,
are very highly upregulated, and that too it is upregulated even in our flooding genotype. But the upregulation is better in the adapted genotypes.
hmm.
So that
Okay.
that made us to like come to this conclusion that more number of nutrient uptake genes are upregulated, and these nutrient uptake genes and mechanism may contribute to the aerobic adaptation.
Okay. And for my own interest, I'm also asking, maybe it is helpful to others because now you have published many paper and one directions. That is the beauty that you are not focusing in too many things, but all are related to roots. So it's a very good research. Indeed, is a good research in that way. But if for example I have multiple sequence of resequencing, means 20x or 25x genome sequencing. So if I wanted to check any locus which
Mm.
is dedicatedly related to the root structure or maybe nitrogen utilization and all, so this information is now available from your lab,
Yes.
or means it's a more accurate because we are not in that field, but we
Mm.
are wanted to interest that whether it happen or what is the happen in the root. So
Ye-
this type of specific locus,
Yes.
if we wanted to check, or specific gene sequences if compared between the 10 or 12 varieties,
Yes.
so it can be done.
Yes, it can be very well done. We have on
Okay.
chromosome number 2 and 12, as I had told, for
Hmm.
mutation mapping also related to root and root volume. So this couple
Hmm.
of genes we've also done the allelic mining for these genes. And
Okay.
CASP is also one of it.
Hmm.
That because it is correlating and co-segregating very well with our roots,
Hmm.
that presence of allele can also be checked in. And we have also developed one allele specific primer that I did not show here because
Hmm.
we have done it already with 10 to 12 genotypes,
Hmm.
and it is yet to be like uh, but it is like far more reliable that we can check that's allele specific marker and check the what is the amplicon length also. So if we are getting one particular amplicon length, then it may be better for the root system like that.
Okay, so it can be easily screen multiple varieties in a short time. Just a PCR. Just we need to do the PCR, and there is a high chance that this variety has lead to that. Okay, so it's a
As well
great.
for resequencing also, because we have mapped now all these regions,
Hmm.
these regions can only be checked in the resequencing.
Resequencing. Yes, my purpose is that because I have the resequenced. Because in ILS, in our institute, the sequencing is we have a Nova 6000 sequencing, so we are very used to sequencing and all. So yes, we have we for my one of my work, I resequenced multiple varieties, and now I'm looking for different thing. But now it give me some idea, so we can talk about in future that.
Yeah, apart from this route related to this, we have for other factors nitrogen, for nitrogen also, low nitrogen,
Hmm,
low phosphorus also from other R groups in
hmm,
biotechnology. We have already published certain research articles on GWAS as well as for the SNP, SNP
okay.
genotyping. So
Hmm,
those things can also be checked simultaneously
hmm, hmm,
to
hmm,
get a better understanding of what exactly is happening if it is a nutrient use efficient genotype.
hmm. Okay, so it can give us some idea about the efficiency of nutrition. So we can plan the agriculture practices accordingly.
Yes.
Okay, so in future, because I'm discussing it with you, so people or audience can understand that what is the actual benefit of such studies. So it's not only the root structure or root architecture. Now it maybe in future in a very low amount of fertilizer we
Yes.
can grow these varieties, or maybe we can use them as a direct seeds method.
Hmm.
So what do you think that direct seeding method is more economically viable or more challenging in that way? Means is it now we are ready to direct seed method?
Yes, yes, we are ready. Like at
Okay.
least all our Rice Institute and all that, we are really working on it. In fact, we have specialized projects on DSR right now which are running because it is economical in the
Mm,
way because it saves some transplanting cost
mm,
and labor cost and other thing. Challenging because at the same time we have to very much have a specific breeding approach or specific focus that when we are doing direct seeding, it should be weed competitive, it should have a
mm.
better vigor because when it actually germinates and come up, it should come up before the weed. And
Ah,
it has a
yes.
peculiar canopy. It should have weed suppression ability. It should have a high vigor so that
Mm,
it can compete with other plants. And then better roots also because in the aerobic condition, that anchorage also should be that much proper
mm,
that it
mm,
should properly establish itself and then grow. And along when it grows with that limited water condition, the genotype should also be high yielding because without yield it is very difficult to correlate anything. So with all these things it is challenging because now we are specifically developing such breeding pipelines or segmentations are being done for
mm.
different segments that okay how we can do direct seeding with minimum moisture or aerobic condition maintenance.
Okay. So, yes, and I just go through the few questions of students, like one student, one participant. I cannot can the students is the interested. Their interest in case of salinity tolerant varieties and all. So, what should be the initial number of varieties they have selected for the starting a study on the salinity?
Salinity, if you, or any trait per se, if you want to start with genome-wide association mapping, if
Hmm,
our approach is GWAS, then at least it should be 120 to 150. If it is a
okay.
difficult trait, sometimes if we come on to 98 or 100, that is also all right. But we should have that diversity which is present, even if it is salinity or any other trait. When we are formulating the panel, it should represent different kind of diversity, geographical diversity, or genotypes, or the parentage of it should be diverse. If you want to focus only on the molecular mechanism, and we know that this one genotype has a better tolerance than the other, and only molecular mechanism has to be targeted, even 2 to 4 genotypes are good enough to start with those kind of studies.
Hmm,
And if you want to map the genomic regions for salinity tolerance, then it is better to take one donor which has a high salinity and one popular variety or a genotype, then cross it, then go for biparental mapping population development and map it in the F7 or F8 using molecular markers or SNP, SSR, anything.
So putting a known control is important. Like Pokhali, I think
Yes.
maybe the Pokhali is one of the site. So yes, it gives the confidence and also gives some understanding that okay, we are in the right directions and all.
Even certain land races in that
Hmm.
case, if anybody is interested in those kind of studies, they can
Hmm.
take into consideration land races also.
Okay.
Because
Okay.
in India, we have so much diversity and
Hmm.
rice is grown everywhere.
Hmm.
Land races can be collected or farmers' varieties sometimes. That will also add on to our understanding how this relates or what is a molecular mechanism or
Hmm.
genes like that.
For farmers varieties, just I wanted to know that because it's developed by them or it's maintained by them for multiple generations in that.
Mhm.
So when you receive any farmers varieties, what should be your first aim, that whether it is matched with your databases or is really a farmer?
There is a complete different procedure for that.
Okay.
For farmers identified variety or when we are using it for our research,
Hmm.
there are certain guidelines and we have every institute has an ITMU which takes care of this. Technology units will take care of which germplasm we are using and we are procuring.
Hmm. Okay. So there is no chance of mm, mm, means scientifically it can be incorporated in that way
Can be
that
copied.
that is used in that. So one now it's okay. We come back here once again. We go for few questions related to on your journey in science. So already you have mentioned few of them in your talk, but for sake of our interview, we maybe it's look like a repetitions, but let's start in that way.
Sure.
So your work is totally focused on root system architectures for water use efficiency in rice and also the nutrition efficiency in rice. So what do you think that this field of research means root system architecture uh, uh, can be improving our water efficiency, or it can minimize the shortage use of water in rice cultivation, because rice is a very water demanding cultivation process. So what is the future of your analysis and all?
Yes, it reduces water. That is there. And for this reduction of water only, we are like one part of research we are doing on aerobic rice. That is a limited water condition. How best we can grow rice with
Hmm.
minimum availability of water. So we have already shifted this or we are already into it, the research of it, like how it is. Because water conservation, yes, where water is available, definitely it is not an issue at all. We can go with normal rice cultivation, rice genotypes or popular varieties cultivation. It is a problem of the future. What we are expecting is rainfall will be erratic or sometimes it will rain, sometimes it's not exactly that time will match with our transplanting or sowing or something like that. So for all those things, direct seeded rice or aerobic rice maintenance is a very good option. It is water saving technology and many of the states are also adopting right now, but it is very much
Hmm.
specific to the pockets according to the availability of water or whatever. Now germplasm or varieties also are being released in that line. They are
Hmm.
specifically meant for those states or for those pockets for cultivation wherein such kind of issues are prevailing. So we have to take it in such a way that yes, water conservation is required, but we are like all in all ready with our armory that okay, this is a condition, this is a genotype which requires less water and it has a high yield. Okay, we can recommend that to that particular region.
Hmm. Uh, okay, ma'am. Second, the next questions is that uh, so it's a similar. So I just gave that questions, and Yes, and one we just received a very interesting questions that's why I forget. Before I forget that, I ask you that if one student is interested to uproot a plant from a field, there is no plan of doing the research of root architects. Now, she or he interested that let's take the root. So, what will the process of uprooting? Because it's a very common, maybe very general questions from others feel that how we just pull off or
We will not generally directly pull out.
okay.
We are doing it from the flooding field. It
Hmm.
has to be done very carefully because already water is there. So
Hmm.
generally, presence of water only we will do uprooting because it eases that water and moisture will ease, and we'll generally remove little bit soil which is adjoining soil, because in rice also we'll follow certain spacing.
Hmm.
We
Hmm.
are doing when we are growing it in the field. So from the field, we just very carefully we do the phenotyping, remove soil, then little bit we do some mechanical thing process like moving it in all the directions and then slowly uprooting it. Because
Hmm.
uprooting has to be very careful. Sometimes we take spade and remove the adjoining soil also without disturbing the main plant. Adjoining soil maybe like 20 centimeter or something like that we will remove, and then we will do it by hand also, and very carefully we'll remove by doing certain mechanical, giving it a in action, some kinetic actions to it, and then pulling
Hmm.
it up, not direct, because direct pulling will always cut the roots, and
Hmm.
very carefully only it is like uprooted.
So it's one sort of archaeological survey type of
Something like that. Because
yes, to dig the fossils and
instruments of Shablomics or any other things, because
hmm.
those instruments which are like very big, it's really very tedious to go with hundreds or 200s, because we are not doing one plant phenotyping doesn't mean only one single plant.
Obviously. Hmm.
Replication.
Hmm. Hmm.
The field, either the field has to be replicated, and even the number of plants which we are selecting should be minimum 3 to 6. So accordingly, we are planning it and then doing.
Yes. And like your team already aerobic adaptation mechanism in rice, there you are also working that how it is aerobically adapted. So um, can you just form what I can say, just give us some takeaway message on that topic that what is the key features of this mechanism and how can it help in developing drought tolerant rice, because drought tolerant rice is a very interesting topic.
Now, aerobic condition here, as I mentioned earlier also, it is limiting water condition. It's not the complete withdrawal.
Water with of water.
Per se in drought, we are completely water is withdrawn.
Hmm.
So, in fact, for developing aerobic lines, some kind of drought tolerance is required. But
Okay.
for drought tolerance, when we are doing breeding for drought tolerance, there should be very specific in terms of roots. If I say the root angle is very important, which will allow it to grow deep down
Hmm.
up to a level of like 30 to 40 degrees steep, so that it will take, and the roots also should go down. The roots should be heavy enough, the surface should be heavy enough to take out or to access the water which is available deep down, even 60 to 70 centimeter. So the root architecture should be governed like that. And for this, already known genes are there, like DRO1 is a QTL which is there, which is being deployed. We have yield under drought QTLs, QDTY QTLs, which should be deployed when we are doing breeding for drought tolerance. But what here we are doing is for aerobic condition or dry direct seeded condition, where minimum water is there, it is present. So same in our aerobic mechanism also, we propose the pathway like aerobic adaptation mimics drought. But
Okay.
what genes we are getting upregulated under drought stress, we are
Is
not
it different? Hmm.
getting it here. We
Hmm.
are having upregulation comparatively high for nutrient efficient or uptake genes, which increases its efficiency or the better system to absorb. Nevertheless, we are having DREP genes, it is expressed. Even one experiment we had conducted wherein DRO1 gene expression we tried to access like how is the gene expression, but under aerobic condition it is not that highly upregulated as it is in the drought condition. So for aerobic, the traits what we follow specifically when we look into the root is that it should have a early vigor. Like one genotype, in the fourth day, it is the mesocortical length is maybe like 5 mm. So if it has to be better in the establishment, the genotype which can grow 10 mm mesocortical is better along with the root. Definitely root length, root volume is there, but establishment also is first critical point for aerobic rise.
Okay.
And drought screening also, when we do for drought tolerance, is also at the seedling stage drought tolerance. In some of the conditions, we will have drought tolerance for reproductive stage, because reproductive stage would exactly correlate with the drought condition. So initially it is all right, but drought condition, what happens? So in that way, the breeding approaches can be managed for drought, and for aerobic, it can be managed like this.
Okay. Okay. Understand. And one study you have also done, and we are also did that same study because I think we both are understand how important is selections of a internal control or reference gene or housekeeping genes for the QRT data and all. So for rice, that is beta tubulin, actin, these are very famous, means these are well known. But so in your study, because I'm not read or gone through your paper, so what is the beauty of your three reference genes? Means it is very specific to root system related things, or it's the variety you have selected where it work? Means is there any reason that why you are not using them?
First of all, the thought which we gave is, since we are starting with RNA-seq and transcriptomics,
Hmm.
and the molecular study, QRT gene expression is very, very important and crucial for validation.
Hmm.
So we thought for our validation, why not, instead of taking other genes, we get a perfect internal control? Because if we go through all the literature, it is always preferable. Of course, actin and tubulin are used, but we thought, like, why not, when we are going to work for the next couple of years on this, why not to have our internal control and make it doubly sure for accuracy and reliability that these are a set of genes? Because we tried initially taking one or two genes, then the references and the golden standards also says that a set of reference gene is always better when we are
Hmm.
exclusively working on reference genes validation exclusive. And we were thinking, like, okay, once we are doing with this gene, again I should take, because it always has to be panel. We are working for
Hmm.
aerobic or limiting water condition vis-a-vis to popular varieties. So every time popular, and if we have to be like always confident, we should take a big panel. That is
Hmm.
what is advised, especially for rice. We are doing in 2 or 4 genotypes. Okay, try the same thing on a bigger panel of 10 to 15 genotypes and see that whether it really works or not. Because always we have to eliminate the doubts what we have. So for that, we thought, like, okay, 10 or 12 genes were already reported. So we have also taken it from the literature. We selected 10 genes, and then we have done it in two genotypes, contrast genotypes
Hmm.
at 4 stages in the roots and shoots. So it
Hmm.
is a big panel of 48 tissues we took
Okay.
total, and then validated. And then we have used that our very standard golden approaches of how many pairwise genes are required for every experiment. Then
Hmm.
what is the stability value, V value, M value with genome,
Hmm.
and using couple of another 3 to 4 softwares, which
Hmm.
tells us pairwise, like, okay, two reference genes are good enough for our experiment based on its stability and statistics. So like that, again, we standardized for our set of genes. Now we are using the any two genes out of these three, it is giving us a good CT value,
Ah.
stable CT value. In
Hmm.
fact, the cycle threshold values, when we are using with these endogenous genes, it is stable. It is stable across the conditions also, and across the tissues. Sometimes point differences are
Ah,
there. That is
it's okay. It's must be there. Hmm.
but all in all, we are getting good results with using tumor protein homolog and expansion protein and membrane protein as well.
And their performance is better than beta tubulin and such.
They are like stable. When we are using these 2 or 3 in the one, what is like expected out of an endogenous gene, like without any kind of stress,
It
should
should be stable.
stable, that we are getting results.
Even for endogenous gene, it should be stable also in the stress.
Yes, that's very
Otherwise,
important.
how we could measure the stress if it
Yes.
is also changes?
Yes.
So yes,
That was an exact idea like water stress, like water we are irrigated and aerobic. Aerobic is limiting water. Okay, let's get, and we are using always tissues also different.
hmm,
In almost all our studies, we are using shoots as well as roots.
hmm,
Some other manner. So that we thought like for reliable and accuracy, we will go on with this set of genes, and this from past 5, 6 years, we are using for our experiments this
hmm.
gene for water and aerobic and DSR and other things also.
Same idea because we also feel the same thing, and when we change the different developmental stages, then we understand that the previous genes are not working.
Okay. Yes.
Even my own previous paper is not working in the older age of plants. So I revisit the all things, and we also start for 44 genes and came up with the ultimately two genes, and all the possible six cultivar multiple generation, different tissues, different conditions, and 10 different stress, one biotic and 9 abiotic. And we are published in Plant Stress Journal uh, last year. So now we have we are confident. The same reason we start, and we also took the all the known that time known published and popular housekeeping genes, and we point out that it's failed here or it's failed here and it's failed here. But yes, out of two, one is already previously reported, but reported within the 3 stress. Now it's reported in 10 stress. So yes, it's a very basic necessity actually when you study the gene sequences and all. So yes, takeaway message for the participant is selections of housekeeping genes is very important. Otherwise, few days, few year or few months after, someone claiming that your result is not reproducible because of their using different housekeeping gene. So word housekeeping genes you are using, it doesn't matter, but it has to be a housekeeping gene characteristics. Otherwise, it's very difficult to study such minute changes in real time and all. So yes, again, it's a good uh, help for participants for this discussions. So now, just little bit.
And also, anything is there, they can write, we can share papers, articles,
Yes,
everything.
that is my next question actually. People are asking about your softwares and all. Although you have mentioned a slide where the mention all the softwares and whether they're free or not, and with the reference. Last column was the where the software is published. So yes, sometime paid version of software gives some added advantage, and maybe this is the reason why it is paid.
Yes.
And sometimes it's free software do the best thing. If you do multiple using multiple free softwares, then you close to the paid versions and all. So in your field of study, so you already mentioned in your talk, but
Yes.
the repetitions. So if a students from a college who just interested to let's check the root, not even rise in any plant and all, because we wanted to people have come to the root biology and all. So microscope is there. So imaging is there because you, people are find the very beautiful images of previous speakers and all for the microscopy of roots, but the measuring of roots, isolating of roots, then what should they do first? The collect the root, then wash it, then measure it, then software. So just 4 5 steps I wanted to know from you.
very first thing is very careful extraction of the roots from whichever media we are using, whether it is soil, agar, or hydroponics, anything. After that, we have to wash it very, very carefully. Either we can dip it for some time. If it is soil, what we generally do is 10 to 15 minutes, we will like just soak it in the water and then take it out so that maximum soil will be getting out of the roots, and very carefully it has to be again washed. And we have a very, very good expertise here. Like one of our boys there, Jagdish, he's a technical one. So he'll take it very properly. And we are also like all assisting there and sitting that it has to be very careful, because if once extraction and the harvesting is perfect, then we don't have any issues for the further thing. If the root mass, because in the irrigated condition or where water is there, if the root is too big, we are dividing it into half and then doing all the parameters into half. After doing this, we are measuring with a measuring scale, the root length. Under aerobic condition, till now, we have got 2 to 3 lines of germplasm, which can go even up to 60 to 75 centimeter down. So
Okay.
along with that, proper scaling, and then we are photographing everything properly. So after scale, like doing a measurement with scale, photographing, we are using water displacement method to understand the first volume, the basic by Archimedes principle, what we use, like just dipping and then understanding what
Mm.
is the root displacement method. Then that again we are keeping in moisture, again washing 2 times and subjecting it to a scanner software, which is WinRiso. We have a pro version of it here at our institute. We have a basic version also and pro version. So it is actually an instrument which comes with a dongle, and it comes with a scanner as well. So we have to wash very thoroughly the roots, put it in the scanner, scan it once. The scanning takes approximately 60 to 70 seconds. And then select that area peculiarly where it will again scan. And there, when we are doing root scanning, we have to be doubly sure that every root is separate. So we are using the needle brushes and then doing maximum separation of the roots. That's what, if the root is like too heavy, we can divide it into 2 parts and then separate it properly so that the scanner scans every root perfectly. And after scanning is done, we are analyzing that, and the software gives us again the volume. It also gives the length, but basically we are using it for root diameter and the number of root hairs which are present. Just excuse
Okay. So yes, is a very long webinar and talk. So just we try to finish one and two questions. First one, I wanted to know that um, how you came to the root architecture and this study means how it motivate you, and what is the what is the trajectory of your career when you enter this thing?
So I actually joined ICR in 2014, and after our all preliminary professional trainings and all that, when I came to Rice Institute,
Hmm.
already our institute is like a very premium institute. Our biotechnology and crop
Hmm.
improvement division, almost all aspects were being covered in each and every step,
Hmm.
like for yield related traits, and for everything. Like as I told in my slide also, so we were working for everything. So we thought like, what else is now
Because
remaining
very few are
for
left in rice. Everything is done in that way.
like to go or digging deeper into which trait we should follow
Hmm.
or known. So with all our mentorship from everybody in our department and during our different presentations, we
Hmm.
thought like, okay, little less explored thing. The
Hmm.
roots are not explored or not understood to this level of gene mapping or molecular mechanisms
Hmm.
as the other traits are. So that time when I joined, we had very good mentors, our directors. In fact, the earlier work I had done with Dr. Anand Kumar sir, and we
Okay.
had all our heads, all our seniors,
Hmm.
Shishu sir, Dram sir, Nija madam, Satyendra sir, everybody motivated in such a way, even
Hmm.
our head of the department that time. So everybody told, okay, this is a good trait to start
Hmm.
with or
Hmm.
to begin. So I thought, okay, we should see the unseen first and go
Exactly.
deeper into. From that point, starting from phenotyping or standardizing from 10 to 20 genotypes, at least now we are at a condition to do it in hundreds. I cannot say we can do more than 400 or 500 like that.
Obviously, but gradually it can be increased in that.
100, 150, which are like manageable within our level. So in that way, I started working on roots.
So, have you ever tested the known varieties? What happen when they are grown in hydroponically? Means,
Yeah,
is
or
there? Hmm,
maximum the popular varieties also we are keeping every time
okay.
for all our studies under aerobic or anaerobic. I can give you one example. There is a genotype called Moroberkan, which is
Hmm,
used for drought tolerance and other thing. But when we tested it here, or when we evaluated under aerobic condition, that some genotypes which are better, they may have a very longer deep root or a longer length of the root, but
hmm,
may not be having a perfect thing to express in such a way that it is adapted to that particular condition.
okay. Hmm,
That condition, that point, in the whole our study, we have tried to focus like what is our best.
hmm,
Every genotype is good only. It is nothing like flooding adapted is not good, but it suits the suitability and the condition, the stress what we are imposing,
hmm,
or how it deployed further. Specifically for roots, we are trying to check it out, but every time we are keeping controls like
hmm,
the mega varieties we are keeping, or when we are checking it under aerobic condition. Yes, it gives better. It is not like even if it is flooding condition, but we are evaluating the genotypes which will be better than those checks under that condition
hmm.
of nutrient deficiency. But we are every time we are checking the popular varieties, or checks are also there with us.
Okay. So, for this, it will be the last question, and that's for this session. And for participant, I just shared the feedback link, and this same link is for your e-certificate application. So, check it and submit your feedback. And this link will be available for 3-4 hours. Don't worry, and use the same email ID. And you got the certificate after few days when I get the time, and I'll prepare it. So last question, Kalyani ma'am, that what what is the takeaway message of new budding scientist or plant scientist? What should they do? They are the combined. I wanted to know the informations regarding their study style, their AI using skill, their actual field experience, their writing skill, everything. This is the last question from our bioengineering webinar and all. And thanks, ma'am, for accepting this invitation. And I am sure your talk will be very, very enjoyable for others, because it not only the result, it also has the background information, descriptions, and all what he want from you for the sake of people those are not from this field, but also those are the learning. For me also, it's a very good understanding about the roots and all. And yes, I admire your patience, challenge, and last 1 decades of work on that field. And maybe one day it will very helpful for farmers and selecting the varieties, or for projecting that what happened when the nutrition fluctuations is that way, or zero fertilizers or minimum fertilizer use in rice cultivation. Thanks. So your answer.
So my answer would be, for the young budding scientist or the students or research scholars, please really take interest with whatever you're doing. Don't get driven away too much for this AI and other things. AI is useful, as you said, the first question is like AI. We can do our best with whatever we are doing first. We should harness complete our energy. We should foster some kind of innovation with a complete uh, dedication of sticking onto it. Initially, we may get like everybody will advise us the same thing. That is what I'm presenting also, but we have to stick on to it, whether it is result or no result, or result is actually a result. Even if we are not getting something, at least we have a way to know that because of doing this, it is not coming. So if that kind of attitude we are keeping from day one, definitely we land to a perfect thing, what we want. And we should always foster innovations. Like there may be a reported thing which may work, but if you are finding like, okay, this is not reported, and especially in agriculture science, this is very evident because it is so diverse. Crops are diverse, our systems are diverse, our ecology is diverse, geographical things are diverse. But whatsoever expertise is available and whatsoever collaborations are available, it is always good for collaborative research. And we, like exactly we say, no, we can foster innovation. I like coordinate or collaborate for expertise, then take the, even the infrastructure collaborations are very good. See now, whatever we have been like showing, or I talked for half an hour or 1 hour or so, so many collaborations are there, so many funding agencies are there, lot many people are involved. Even if I am presenting, there are at the back 40 or 50 persons who really worked in one or the other way. And we are also using the all India coordinated research projects. We are giving somebody for phenotyping because expertise actually matters here, and along with the infrastructure. So anybody who are using for the first time, or they are into the new system, developing their research ideas, so we can approach your seniors. We can go with collaborations, not getting like too much distractions. We can follow even a single method, even if it is failing one time, two time, definitely we'll get some other idea. So ideation is also much more important along with the innovation. Like some trick somewhere we'll get. I can give an example. For this long non-coding RNAs also, when we were analyzing one of the data for transcriptomics, two or three IDs initially, I was getting completely confused. Like these IDs are not matching to any of the databases, neither with RefSeq or neither with any mRNA. Then what they are doing exactly here? Then since when I joined, I was given some transcriptomics data for which I'll be like highly obliged to all our seniors over here. So when I saw those data sets, in peculiarly one or two data sets, I could not match these transcripts or the genes with any of the databases for mRNAs. Then I was thinking, no, something is a problem, or something would have gone wrong somewhere. Then again I started reading, okay, we used ribo0 for a mRNA extraction or what, everything. And then after along 1 or 2 months of searching all the database, I got like incidentally or coincidentally, these are the long non-coding RNA which are expressed in the data. And that was an idea which clicked me early. Almost 2-3 months I was thinking like, because something is wrong, I am getting this 10-20 transcripts from somewhere, or it may be contamination. And moreover, with when we are working with roots, it's like so much like other microbiome,
Others microbes.
so many. In spite of our methodology right, the procedural right, or when we are isolating, or when we are giving for next generation sequencing, everything. And after that I got to know, oh my god, this is like something called long non-coding RNA. So it actually enthusiasmed me that okay, something is there which I was considering 3-4 months, I was considering it as a wrong thing. So like that, if we come on or to stick on to one point, definitely we'll get. And this is advice, not, or this is like very friendly, everybody will be giving us the same advice, but this only I want to give. And second, on AI also will give you correct answers if we approach and give the query correctly. So for
Exactly.
giving
Prompts.
query correctly, what is our problem, what we want exactly, and what can be the reasons. So for that, AI is very good, it helps, it is good, but we should understand and write to ourselves first what exactly our issue is, wherein we have to concentrate, what we can do, or what are the probable approaches by which this thing can be done. So that will be a better or a holistic approach like that. So
Exactly.
and all the audience who are listening to me, I'm thankful so much. And any queries, anything is required, we are all like in touch with WhatsApp or email or anything. So thank you, Team Biogene, for giving this opportunity
Yes,
also.
it's our pleasure, ma'am, and thanks for coming so nicely, present your work and the descriptions, and it's extensive actually. By in 2 hours is I know what happened for the in speaker ended, because it's a very extensive one. But thanks for participating, and hopefully we wish you for a better future in science. And yes, we are going to uh, getting advantage of your studies. And yes, from bioengine website we put your all information, so means your different social medias or LinkedIn, your official websites, and also people can connect to you, and maybe they are entered to the biology, which is not sin in what I can say. It's a very few people are working on that, so
Yes.
should be nurtured in that way, and we are very happy.
Show opportunity to also inform that
Hmm.
our institute, we have lots of germplasm. We have a complete set of genotypes, which are very
Okay.
well adapted genotype for conditions from abiotic, biotic stress. And as well as we, along with the genotypes or along with the varieties which have been released, we also maintain the germplasm, which we have registered with NPBGR New Delhi. We
Okay.
have our own microbial isolates also. So those who are really interested to collaborate or to
Hmm.
use our lines or collaborate on any kind of marker study or anything, we are open. And then we have all the publications. Those who want, they can access our publication from our website, as well as they can directly write to us. And
Hmm.
we are also on social media. Everywhere we are following. Any new publication, we are giving it on Facebook, Twitter, so that anybody really interested, they can take a
Yes, they
deeper
can
look
connect.
into,
Yes.
and they connect. And
Hmm.
especially for the students also, because like as a member, or now I'm talking as like from our institute side, as an
Hmm.
ITMU member also, I invite all the students to do 3 months internship or 6 months internship, whichever they are interested into. So we are
Hmm.
into that. But really, those people can approach those who really want to work hard, because
Yes.
this is
Yes,
rice.
agriculture. It's
uh,
agriculture, and it's not so easy. Yes.
Field plus lab. So
Hmm.
this kind of integrated study is really required to
Hmm.
take to up to a level of product as well as basic and applied research.
Okay. So for internship, information is available in your institute
Yes.
website, or
Yes.
directly contact to scientists. Okay, information is there.
We have a special academic committee for this,
Okay.
and people can connect to our academic committee with all the procedures and other things, with all the formalities and guidelines. They are available on our website also.
Okay. So
And if
people
any,
can complete the dissertations, no? Like their dissertation training and all. Okay.
yeah,
Hmm.
we do
So
visits,
obviously, hmm.
even visits
Obvious.
those fields. So for that also, like, we are open and doing
Yes,
that.
and your institute is one of the premium institute in rice research, is no doubt, and their contribution is not for only one type of research. It's a multiple type of diverse research, not a single checkpoint. Is a good institute, what I can say. And
Yes.
yes, we are happy to help people if they are interested to
Yeah.
connect any of our speakers.
Anybody really connect to us, we are thankful.
Mm. Yes, and that will be more justifications of giving that type of talk and all, that people are connecting, and we are very on this aspect. We are very serious to connecting the people in that way. And thanks, ma'am. It's a great pleasure to you. Maybe in future, after 3-4 years, we will invite you and to the next level of your research in that way. Okay. So
Okay, thank you
bye.
so much. Thank
Okay.
you.
Thanks very much for coming.